everyone, my name is Steven and my student ID number is D202211011740.
Hi, Assalamualaikum. My name is Muhammad Abdul Nabi Aizudin and my student number is D202211011738.
Assalamualaikum. My name is Syed Ahmad Muzamil bin Syed Jufri. My matrix number is D202110993949.
Assalamualaikum warahmatullahi wabarakatuh, and my name is Ahmad Safran bin Abdul Samad. My matric number D202110993959, and we are from group 3. Alright, this is our replica DNA. So we make our replica from clay, ice cream stick, balloon stick, toothpaste, and cable tie. So this is our replica.
A nucleotide is the basic building block of nucleic acid, which is RNA and DNA. A nucleotide consists of a sugar molecule, either ribose in RNA or deoxyribose in DNA, attached to a phosphate group and a nitrogen-containing base. The base used in DNA are adenine, cytosine, guanine, and thymine. In RNA, the base uracil takes the place of thymine. DNA and RNA molecules are polymers made up of long chain of nucleotide. Deoxyribonucleic acid is the molecule that carries genetic information for the development and functioning of an organism. DNA is made of two linked strands that wind around each other to resemble a twist ladder, a shape known as a double helix. Each strand has a backbone made of alternating sugar and phosphate group. Attached to each sugar is one of the four bases, which is adenine, cytosine, guanine, or thymine. The two strands are connected by chemical bonds between the bases. Adenine bonds with thymine and cytosine bonds with guanine. The sequence of the bases along DNA backbone encodes biological information, such as the instruction for making a protein or RNA molecule. DNA replication is the process by which a double-strand DNA molecule is copied to produce two identical DNA molecules. Replication is an essential process because whenever a cell divides, the two new daughter cells must contain the same genetic information or DNA as the parent cell. Semi-conservative replication. In this model, the two strands of DNA unwind from each other and each acts as a template for synthesis of a new complementary strand. This results in two DNA molecules with one original strand and one new strand. Conservative replication. In this model, DNA replication results in one molecule that consists of both original DNA strands, which is identical to the original DNA molecule, and another molecule that consists of two new strands with exactly the same sequence as the original molecule. Dispersive replication. In the dispersive model, DNA replication results in two DNA molecules that are mixtures or hybrids of parental and daughter DNA. In this model, each individual strand is a patchwork of original and new DNA.
I will continue with the similarities between DNA and RNA. For the first one, it is composed of nucleotides. For the next one, polar molecules due to their negatively charged phosphate groups. And the third one, both have hydrogen bonds between the nitrogenous bases. The fourth one, phosphodiester bonds between adjacent sugar and phosphate groups. It make up the backbone of DNA and RNA. Phosphodiester bonds are much stronger than hydrogen bonds. It is covalent bonds. Hydrogen bonds are constantly broken to permit for DNA replication, while phosphodiester bonds are hardly ever broken to natural processes. Alright, for the next part is the difference of DNA and RNA. For the first one, the basic shape of DNA is double string, while for the RNA is the single string. The next one, the nitrogenous basis for DNA has adenine, thymine, cytosine, and guanine. While for the RNA, it is adenine, uracil, cytosine, and guanine. For the next one, a type of pentose sugar, where DNA have deoxyribose, and for the RNA is ribose. And the next one, the helical nature of the molecule. For DNA, it is a double helix of two antiparallel polynucleotide chains, while for RNA, it is a one polynucleotide chain twists into a helix and coils back on itself. So, some section of the chain join to other parts of the chain for tRNA and rRNA. mRNA is linear. The types of molecule of DNA. One type of DNA with numerous variations due to the arrangement of the bases, while three types of RNA, which is mRNA, tRNA, and rRNA, each having a different role in protein synthesis. The last one is the row, where for DNA, it is a long-term storage of genetic information sequence of basis codes for assembly of amino acids to make proteins, while the RNA is to protein synthesis. Protein synthesis is the process in which cells make proteins. It occurs in two stages, which is transcription and translation. Transcription is the transfer of genetic instruction in DNA to mRNA in the nucleus. It includes three steps, which is initiation, elongation, and termination. Initiation is the phase during which the first nucleotides in the RNA chain are synthesized. It is a multi-step process that starts when the RNA-P holoenzyme binds to the DNA template and ends when the core polymerase escapes from the promoter after the synthesis of approximately the first 9 nucleotides. Elongation is a regulated process in which a RNA chain complementary to the template strand of DNA is synthesized as RNA polymerase moves along DNA. Transcription elongation is preceded by transcription initiation and is followed by transcription termination. Termination is a highly intricate process for mammalian protein coding genes. First, the chromatin template slows down transcription at the gene end. Then the transcript is cleaved at the poly signal to release the messenger RNA. The remaining transcript is selectively unraveled and degraded.